So this is going to be the overview of photosynthesis. So what photosynthesis is the process in building something by using sunlight with the use of light energy, water, and carbon dioxide. Into it can basically photosynthesis can basically transform into glucose and oxygen, which are two products of photosynthesis. And this process occurs all in the chloroplast, which is the organelle that carries out photosynthesis, converts carbon dioxide and water with the use of light energy. The chloroplast is constructed by the inner membrane and the outer membrane, and the stroma, which is the fluid inside of the whole chloroplast. And this whole process can be broken down into two stages: the light dependent reactions and the light independent reactions. First, we're going to start off with the light dependent reactions, which occurs in the thylakoids. A stack of thylakoids is a grana. The thylakoids oxidizes water and converts it to O2, oxygen. By oxidizing the electrons from H2O, NADP picks up the electrons and is reduced to NADPH. And some of that energy is then transferred by light and is used to make ATP, which is produced by chemiosmosis using an enzyme called ATP synthase. So the products are O2, NADPH, and ATP, and the reactants are H2O, NADP plus, and ADP with phosphate. And again, this whole process occurs in the thylakoids membrane, which is where the light dependent reactions occur. Now we're going to move on to the light independent reactions, which occurs in the stroma, which also happens to be called the Calvin cycle, which is also called the dark cycle. And basically the purpose of the Calvin cycle is to use ATP and ADPH to fix CO2 and produce G3P, which becomes glucose. So how this works basically is that the Calvin cycle takes in carbon dioxide and reduces it into glucose. So here is our glucose. And NADPH is then oxidized back into NADP plus, and then NADPH gives up its electrons, turning it into NADP plus, and CO2 eventually receives those electrons and turn it back into glucose. Then ATP is used to power the process of ATP. ATP is converted back into ADPH and phosphate, and it energizes the Calvin cycle, giving it energy to turn into glucose. And then the reactants are CO2, ATP, and NADPH. And then the products are glucose, NADP plus, ADP, and P. Now I want to go back to the structure of the photosynthesis. So thylakoids are the membrane-bound compartments where light-dependent reactions occur. We already mentioned that. Then the membrane holds photosystems, electron transport proteins, and the ATP synthase. Protons are pumped here during the light reactions as well, and then the Calvin cycle happens again in a stroma where CO2 is converted into glucose. To go back to the Calvin cycle in more of a detail, it can be broken down into three parts: fixation of the CO2, reduction, and regeneration. So starting off with carbon fixation, rubisco, which is a crucial enzyme that is responsible for carbon fixation in photosynthesis, attaches to CO2 to RUBP, forming unstable 6 carbon intermediates, which immediately split into 3 phosphoglycerides. Next up is the reduction phase, so using ATP and NADPH from the light reactions, 3 phosphoglycerides is reduced to G3P, a high energy 3 carbon sugar. And following from that, glucose formation, 2 G3P molecules combine to form glucose, and it is basically the end product of all photosynthesis. Last but not least, regeneration of RUBP. The rest of the 3GP molecules are used to regenerate RUBP, allowing the cycle to start over and continue. And that concludes our photosynthesis overview, also along with light independent, which is the Calvin cycle reactions, and the light dependent reactions. So next we're going to go into more in depth of the light dependent reactions. So the electron transport chain is inside of the thylakoid membrane. So this is our stroma, and this is our inner thylakoid space, all of this area surrounding this. And basically, to start off, a light particle is going to hit the inside of photosystem 2, and it's going to excite these electrons. Also, photosystem 2 is also named P680, and I will mention it why they also call it that in a bit. After chlorophyll loses electrons, which those electrons are then flowing to an electron carrier called plastoquinone, or PQ for short. Chlorophyll is going to take those electrons from water, so we're here now, H2O, um, and it's basically going to oxidize and turn it into oxygen gas, and it will basically give off 2 electrons and 2 hydrogen atoms. So the reason why PS2 is also called P680 is because that's the wavelength of light that the highest absorption can take, and it's 680 nanometers, 680. So then after this, PQ is basically going to carry those electrons into the cytochrome right here next door, or B6F complex, and as the electrons pass through that complex, it's going to pump protons from the stroma, that is from the outside of the thylakoid, into the lumen, so inside of the inner thylakoid space. So it's going to go from here all the way over here. And because of that movement of the protons, this will produce a concentration gradient. So then electrons from cytochrome is going to move into the peripheral protein called plastocyanin, which is a copper-containing protein, and then will transfer the electrons to photosystem 1. And then the electrons have already lost basically a lot of its energy. So in order for those electrons to gain more energy, they have to be struck by another photon of light. So another photon of light is going to hit right into photosystem 1. PS1 is also known as P700, which is the maximum absorption at a wavelength of 700 nanometers. Since that photon of energy struck PS1 and now the electrons have more energy, then they would be moved into another protein called photosynthetase, which is an iron sulfur protein, and they carry electrons to an NADPH reductase. From here, NADP reductase would basically reduce NADP plus, and the electrons are going to leave and will meet up with NADP plus, which will accept the electrons along with the H plus, and will be reduced to NADPH. And then last but not least, these hydrogen ions are going to be able to flow into ATP synthase by a process known chemiosmosis, which is an enzyme going to rotate. This enzyme then is going to rotate and will continue to combine ADP plus phosphate to make ATP. So it's going to combine these two in order to make ATP. And that is the process of the photosystems, basically the light-dependent reactions.